over the introduction, and one of the main focuses or impetus for this paper is COVID-19, which is the reason we're all meeting virtually today instead of in person. So COVID-19 is the coronavirus disease 2019, which is caused by the SARS-CoV-2 virus. It was first reported in Wuhan late in 2019, hence the name, and rapidly spread across the globe. So as you can see from this figure from the Johns Hopkins COVID-19 tracker, it is currently infecting over 188, okay, thank you. 188 countries with over 29 million confirmed cases and 926,000 confirmed deaths 5 days ago. These numbers have increased. Most of the deaths are located in 4 countries, which include the US, which currently has the largest category at least 6.5 million confirmed cases. But COVID-19 and the resulting is not the only coronavirus disease that humans have dealt with. The first one that most people will probably be aware of was the original SARS, which emerged in 2002 in China and had a case fatality rate of 11%. The other major coronavirus outbreak people will be aware of was MERS, the Middle Eastern Respiratory Syndrome. This one emerged in 2012 in Saudi Arabia and had a much higher case fatality rate. While it's been decreased from the initial outbreak, World Health Organization lists 34% case fatality rate. However, there are still some other coronaviruses that infect humans. Most of these are viruses responsible for some parts of the common cold. There's also one which is a bit nastier and is linked to croup, which is a mix of common cold as an antecedent in addition to some other symptoms. So this raises the obvious question: if we have coronavirus diseases, we should then logically have coronavirus vaccines. And well, that's not completely the case. The SARS outbreak fizzled out, the last recorded case in 2004, and a wide variety of different papers reporting development of SARS vaccines didn't really follow up with any results in the clinical trials or anything besides initial research. MERS had a lower priority due to a low infection number for the events, and there have still been some intermittent outbreaks in South Korea and Saudi Arabia. It's still a lesser priority, though there's still some ongoing research. The other common human coronaviruses are part of the common cold or constituents. And from what I've seen, there isn't a lot of push for developing a vaccine for a virus which may or may not be a particularly nasty strain for one season instead of the other. Together, there's very low pressure for successful coronavirus vaccine development, which is a bit problematic as there's currently a very pressing need for a successful coronavirus vaccine. So we then go into concerns when it comes to actual fast development. So vaccine development measures 3 different traits. One, the immunogenicity of the actual virus, making sure it causes a useful immune response. There's also concerns about the safety of the virus, making sure that there aren't significant adverse events or other concerns when doing the vaccination process. Third component is a measure of efficiency, which is finding the optimal dosage between the 2 components that will make sure that it's widely effective in as large as humans as possible. So one of my lab mates, I'm not sure if you can see all of that, it constructed a table of SARS of COVID-19 vaccines that are currently in clinical trials. And most of these, as you can see, only entered clinical trials sometime earlier this year. And many of them are in some form of phase 2 or phase 3, with 3 vaccines in phase 3 when this table was made in August, which is very surprising since normally clinical trials take several years. So this represents a massive increase. In addition, there have been the US, as you may have aware, have been trying to push for successful vaccine development optimistically in midsummer of 2021. I'm ignoring certain other comments of it being earlier. And we have the case of the Russian Sputnik 5 vaccine, which is already announced as going to be released to the general public, despite already being in clinical phase trials. And before I begin, I want to discuss some of the ontology and database that we use to collect and help annotate the vaccines. So VO stands for the Vaccine Ontology, which was an OBO-compliant ontology already developed by Alva's lab. There's also VIOLIN, which is the database we used. VIOLIN stands for the Vaccine Investigation and Online Information Network. It was released back in 2008 with over 4,136 vaccine candidates. These are candidates that are in research trials in clinical stages and ones that have been licensed as fully functional vaccines. There are 15 coronavirus vaccines. When we submitted this paper, this number was increased closer to 70, which is why I included an asterisk, but try to be consistent with the presentation since we have been adding more between the submission and this presentation. There's 218 different pathogens, and all the curation is done through manual review of reviewers, most of which are part of the actual heat group lab or worked with us before. So for the short workflow diagram I have here, we queried online for a variety of online sources for new articles relating with coronavirus vaccines. And if we find some vaccines that showed some immune response, we added it to our initial Excel file. We annotated using ontology. We made a note between preprint and peer-reviewed sources before dividing it into terms that we then analyzed using SciPy module and some statistical tests, and also those that we uploaded into VIOLIN and then included into VO. So again, going back onto the main points, the main inclusion category was that we have a vaccine and it shows that it has some published immune response event with the actual vaccine. If there were follow-up research articles which mentioned adverse events, as part of that, it was also included as part of the information, but the initial criteria was a source describing immune responses. So, which actually removed quite a few SARS vaccines and a bunch of SARS-CoV-2 vaccines that were still under development and didn't have results published yet, which made things interesting. That was the other reason why we still collected preprint sources. We made sure to separate them out from the analysis and tag them as appropriate preprints, but we still collected them in anticipation that most of them will become peer-reviewed sources in the near future. So for our actual ontology terms, we used vaccine ontology for most of the immune response annotation, and we used the ontology of adverse events for the adverse event notation. And finally, for the VIOLIN curation, we looked over the information we added to a database. We then reviewed it before making any approval for the information. Preprints can still be added to VIOLIN, but it was a conscious decision not to approve them until they have a peer-reviewed source. Okay, how am I doing on time? Because I know we started 10 minutes late. Okay, I'll continue. So for the immune response and adverse event analysis, we split vaccines that had information during the research in the research phase, and those were in the clinical trial phase. 5 of them, we had useful clinical trial results of the ones that we collected in time for the paper, while almost all of them were exclusively research sources. We tried running basic statistical analysis between the distribution of the immune response and the adverse event types, sorting by those caused by the vaccine, those by the vaccination process, and we used again, I mentioned the SciPy module to run a one-tailed Fisher's test to see if there was anything that was statistically unique. So here we have an ontology representation of a COVID-19 vaccine, AD5-nCoV. We classify each vaccine by the microbe that it targets instead of the disease process in this diagram. And you can see the SARS-CoV-2 is represented through the 85N-CoV immunizes against a microbe. We include the relationships between the vaccine antigen, between the type of vaccine, and mention the components. In this case, the recombinant viral vector role, because it's a recombinant viral vector using the adenovirus serotype 5 vaccine vector. We have a relationship to describe the vaccine-specific immune response and as part of the vaccine, while the actual vaccination process is we have the relationship between the adverse events. Okay, skip me. Oh, and you can see the origin for all the other ontologies that we used down below in the figure. So when we submitted the paper, we had 58 vaccines collected in VIO, and which is shown in this table. So entries on the left are how many SARS vaccines of a given category, MERS and COVID-19. The top row represents the antigens that were used, while the column headers, the row headers, sorry, are used to represent what type of vaccine they were. So the most frequent one were DNA and recombinant vector vaccines, but DNA vaccines are ones that have a plasmid with the DNA sequence for the antigen to be produced. And while recombinant vector is usually a viral vector that's injected into the host, presenting the antigen, and then that's used to trigger the immune response. While most of the vaccine types were already existing in VO, we did add 2 new vaccine types. We have the virus-like particles, which are the creation of a virion with the antigens, but without the actual RNA infection, RNA that's needed for infection. And then we have RNA vaccines, which inject RNA into the host, which will then be translated into the antigen itself. So the most frequent proteins or antigens were all proteins, which is the spike protein, which is responsible for how the SARS-CoV-2 virus infects other cells. We do have some other antigens that target the membrane protein, which is the M, the nucleic acid protein. There were, of course, a few cases where they use a whole virus as the antigen or some combination of the two. So for the immune response is that most of the papers did report some antigen-specific immune response for the actual virus. There was one exception with the M protein vaccine, which didn't actually show something consistent in the immune response, but that when combined with a nucleic acid protein, it actually did provide some immune response against SARS-CoV-2. The other one is that going over, we had a surprising small enough that shown protection through a challenge study. So several vaccines only demonstrated that using the immune response or that there was heightened antibodies that bind to the actual virus when removed in the other case. But they didn't do a challenge study to show much stronger evidence that the immune response was effective. So this was likely the result of issues with the model organisms earlier on, especially with mouse, having issues actually being infected with the SARS coronavirus. Yeah, I'm not sure how much time left, so I apologize for rushing. So common immune responses that happened were the presence of interferon gamma with immunoglobulin G or immunoglobulin A, increased interleukin production. And there was some evidence between different studies that vaccination route provided different efficacy for greater immune response. And there were a few rare cases of reporting some any Th1 or Th2 cytokine response as part of the immune system. And those cases generally tend to match up with ones that had some adverse events. So there were a series of studies that used biopsy female mice. And you can see that For a common set of adverse events, there seems to be a very common pattern of an unbound CH2 immune response, which we had as an adverse event of some lung infiltration of neutrophils or eosinophils, which happened in these vaccines. Most of the non-DNA vaccines had the presence of an N protein, either as part of the whole virus or as the other epitopes, suggesting that there might be some link to specific antigens, which are responsible for producing that, but we didn't have any experience that conclusively showed that property. I would also like to take a moment to talk about a SARS vaccine, ADS-MVA. It is a viral vector vaccine that when first tested in mice, didn't see any recorded adverse events. However, there was a follow-up study which shown the opposite property, where there were several severe adverse events that were ongoing, mostly in the liver, where there was inflammation, greater necrosis, and other damage from there. So when we go over to the clinical adverse events with humans, which we can see in this table over here, which again, it's only limited to 5 due to some of the issues mentioned with the vaccine collection. So each of the vaccines had the number of participants shown below, and the number of participants that experienced an adverse event in each of those rows. Okay, so commonly, most adverse events were mild to moderate. There were 14 occasions with grade 3 events being recorded. These are ones that are considered severe and much more problematic in terms of the human safety. Only one that we saw that was universal to all 5 was injection site pain. However, when we tried running the Fisher's test to look for unique statistically significant adverse events, which you can see with all the values with an asterisk, we found that the mean adverse events were about 5 per through a range of 2 or 9. The most common number was actually 6, and so showing that most vaccines tend to have a unique set of adverse events, which makes it a bit harder to make accurate predictions on what adverse events any future coronavirus vaccines have. So in conclusion, most reported vaccines demonstrated some measure of immune response. I suspect that might be due to the fact that they would only publish if they had positive results, but I have no evidence supporting that assertion. They reported some protection efficacy through the use of a challenge trial and relied on small measures of increased immune response or presence of specific interleukins or viral specific or antibodies. Most research stages reported less adverse events than clinical trials, or this again might be the result of different experiments sacrificing the animal after a short period of time instead of waiting a longer period to observe for adverse events, or it might also relate due to the different focus between research and clinical trials. Again, mostly mild, but some severe. We also saw that using different animal models can include potential adverse events. I know that there has been work with using African green monkeys and other primates as model organisms to get a more accurate reading than the issues they had with mice. And I just want to reiterate that there's still a lot of unknown with coronavirus vaccines that are currently being developed. And I just want to reiterate a word of caution for any future development and keep an eye out for adverse events. Okay, thank you. How did I do on time? Okay, that was a bit long.
Okay, thanks, Anthony. Any question for him?
Yeah, can I have some questions.
Sure, go ahead.
Sure.
Anthony, can you show how do you represent the immune response?
Okay, so for, I'm still sharing my screen, correct?
Yes.
Okay, so right now we have a variety of different VO terms that we link through induces relationship with the vaccine and induces some vaccine-specific immune response. So I know we have ones for the interferon gamma, we have some for the interleukin 2, 4, and I believe 10, and most of these are sorted through one of the earlier ontology terms.
What OAE ontology terms?
The immune response are actually part of VO.
Okay. Yeah, I'm just curious, maybe because immune response is kind of a process terms, are you using something like GO or other ontologies to represent the immune response? And also, besides the immune response linked to vaccine, do you also like represent some participant in a particular immune response process? For example, this interferon gamma immune response certainly has participant interferon gamma. I'm just curious on the part of representing immune response.
Okay. I remember discussing with the lab with taking a look at some of the GO terms. For right now, we don't have further linkages with much of the actual components to the interferons. I know it was discussed, but I don't think it has been implemented into VO when we last looked at it. But that's definitely something we can do to expand the work.
Okay.
Yeah, definitely. I can add something. Yeah, I think, yeah, this is one, but we have not present the immune response induced by vaccines in VO that closely. This is an illustration. But also, I want to point out there is actually some error here in the figure. So the recombinant viral vector role, this is a role is not bound by the virus. It's bound by by the vector. So the arrow should go from the adenovirus serotype 5 vaccine vector. That vector has the role. It's not the vaccine has the role, I think.
Okay.
Yeah. But anyway, yeah, it's just, I feel like, yeah, seems like it's an error, yeah. Thank you.
Okay, so that was a comment and not a question, correct?
Yeah.
Okay.
Anthony,
yes.
so I know there is kind of manual curation involved in getting this data in. And how often do you update the database? So I'm just wondering. So you showed in more than 69 kind of vaccine, entries here. I mean, are they all active in the clinical trial, or some of them have been kind of terminated, discontinued? Are they reflecting the current status?
So for the actual updates, it varies. We did a bit more frequent updates monthly within the earlier months, partially because there was a dearth of information actually being released back in May. And so there was greater concern in getting the information. After that, once the paper was published and we had a larger corpus, we've been moving more onto every 1 to 2 months for uploading into VO, while we're working more on a monthly set of updates for VIOLIN. We haven't updated yet for this month in September, but I plan on doing so, partially due to other ongoing events, but I plan on trying to do a monthly schedule of updates for new terms in VIOLIN. So.
Okay, thank you.